Santa Cruz, where she used her oceanography background to link elephant seal behavior to oceanography. So you'll hear a little bit about that today. She graduated in 2020, and she continued on there as a postdoc, and she helped to write a grant that we're collaborating on, investigating multiple stressors in northern elephant seals. And then she transitioned into an assistant researcher position, and now I think she's a PI or co-PI on 2 different grants. One where they're tagging sea lions in order to see probably how they're eaten by white sharks. It's a white shark study. And then another one looking at, it's a seed grant to study impacts of climate change on breeding habitat for pinnipeds. She's also dedicated to mentoring and teaching, and many of you guys know that. I think she's trained over 20 graduate students in field techniques, and many of those have been mine. She's on 2 of my students' committees, and she's always willing to help out with coding and things like that for my students, so greatly appreciated. And with that, I will hand it off to her.
Thank you, everybody, for having me this afternoon. So the original title for my talk was Successful Strategies for Foraging During a Marine Heat Wave and a Deep Diving Predator. That was actually supposed to be slightly modified, although a lot of the themes are the same, to oceanographic measurements in a marine heat wave and behavioral response of a top predator. So I am going to spend some time talking about the oceanography side in addition to the behavioral side. So for those of you who are more on the predator behavior side, you will have to bear with me through some temperature and salinity and all sorts of those sorts of things. To start with, I just want to talk about the framework that I sort of try and view most of the research that we do through, and this is not just me. This is a lot of the framework that the work in Dan Costa's lab, where I did my PhD, this is where we're coming from in terms of thinking about links between environment, behavior, physiology, fitness. All of these things are interconnected. None of them happen in isolation. And so our group and the projects that our group work on tend to dabble in all of these things. And people like me who come in to study behavior end up doing all sorts of physiology nonsense over here, because it turns out that that's actually really important too, and they're very, very interlinked and interrelated. Coming from an oceanography background, the environment component is very important to me. And it turns out that it's very important to all of the other things about the animals as well. So interconnections between these sort of discrete disciplines is kind of a theme for me in terms of thinking about research. These are just examples of some of the questions that Dan's group, sort of our larger group, is interested in asking. There are many, many others. This was just kind of a sampling of some of the things we've been working on recently and or things that we would really like to figure out and haven't figured out how to answer yet. A major theme in the last about 10 years of the work we've been doing has been using animals as oceanographers, and this has been happening both in the North Pacific and in the Southern Ocean. So most of this work has been done with northern elephant seals, but we've done a similar type of sampling with southern elephant seals, with Weddell seals, big animals that can carry relatively large instruments. And the interest is in really linking ocean conditions to animal behavior when they are out in the ocean, and that's way harder than it sounds, as it turns out. But a key part of it is actually taking measurements of the environment where the animal is when the animal is there. And that's something that is very hard to do with buoys, gliders, shipboard surveys, satellites, because you often have mismatches of scale in terms of either space or time or both. Whereas a seal is in the ocean right now experiencing this particular location and looking for food and deciding whether or not this is where I'm going to stay and forage or I'm going to go somewhere else. So the decision making happens on very small temporal and spatial scales, and a lot of our measurements are on much larger scales. So really trying to move towards getting those scales aligned to answer questions. And we do this primarily by attaching tags to their heads that measure the ocean conditions where they are while they are there. Like I said, elephant seals are great for this because they're big. So are Weddell seals because these tags are relatively large. So we can't, unfortunately, put them on just any seal, although technology is always improving, and temperature is something that can actually be done on a pretty small scale. So that is something that's available on smaller animals. But chlorophyll fluorescence and salinity require pretty large electronics, although these tags are still like yay big. Like when you compare that to a CTD rosette on a ship, like this is not very big, for instance. So for today's talk, we're going to talk a little bit about environment and what I investigated during the marine heat wave in 2014 and onwards. And then look a little bit at the behavioral responses of northern elephant seals to that marine heat wave, if there were any, and what it meant for their livelihood. And a quick, very important acknowledgement. I am using data collected from 2004 all the way essentially up to the present. And that represents many thousands of people hours worth of field work in particular. None of this type of work would be possible without the Costa lab in general having sustained this data set. And having run many of these field seasons, I can tell you that it is a huge amount of effort. And it started actually around the time Gita was a graduate student. So there are many, many people who have contributed to this work aside from me. So a little quick primer on northern elephant seals. We are focusing on adult female northern elephant seals. Most of our work has focused on adult females for two main reasons. From a population demographic standpoint, they are the most critical to keeping the population going and to reflecting population success. Sorry, guys. Not that the males are not interesting. They are very interesting. But the other reason is that the females are much better at coming back alive with their tags attached. And that's also a very key part of trying to do this work. So they're sort of more of the low-hanging fruit of the elephant seal grouping. They go on 2 foraging trips, one relatively short trip in the spring, followed by a longer trip in the summer-fall. Those 2 trips are broken up by periods of fasting on land when the seals have no food resources for somewhere between 4 and 6 or 7 weeks at a time. They go way out into the North Pacific Ocean, typically traveling between 5,000 and 12,000 kilometers round trip, which is a lot of swimming, and that's just the horizontal part. That does not include any of the ups and downs of the diving part. They forage mesopelagically, so primarily are foraging below 400 meters, around or below 400 meters. The max depth that they have been recorded diving to is 1,764 meters, and there is more than one dive at around that depth, so it wasn't like a fluke of a tag or something like along those lines. And 2 individuals have been recorded diving similarly deep. So they are capable of very deep diving, and the longest dives, it's actually 91 minutes. I apparently rounded down, not giving them full credit, but the longest dive recorded is 90 minutes. Actually, I think that's incorrect. I think it's longer than that. I'll need to update that. In any case, in between those dives, they come to the surface for between 2 and 3 minutes to catch their breath and then go back down for another 20, 30, 40 minutes. And they do this over and over and over through their entire 70, 80, 90, 220-day trip. They are continuously underwater, essentially, aside from those 2 to 3 minute little surface intervals in between. It's really quite remarkable what they are capable of. And FYI, I had no idea about any of this before I started my PhD. I learned that elephant seals are very cool as a result of being interested in studying oceanography, but it turns out that elephant seals are really cool. In terms of their diet, which is very important to this whole question of how might they respond to a marine heat wave, they are generalist predators. So they will eat anything that they encounter as far as we can tell, but their diet primarily consists of myctophid fishes as the #1 contributor, we think, and non-migrating squids. Mesopelagic squids is the #2 contributor. And these two food sources are very different. Myctophids are very fatty, very energy rich. Squids, while being much larger, are much more of a protein water type of food source, a little bit less energy dense and rich. And then, like I was mentioning, they fast twice during the year, once for lactation and once to molt all their fur. And during those fasts, they lose huge amounts of their body stores. So about 35% of their mass during their 4-week lactation period, and about 25% of their mass during their 6-week molting period. And in both cases, they lose around 25% of their lean tissue, which is sort of the trigger for, like, you are going into now emergency, your body stores are too depleted, you cannot survive kind of mode, fasting mode. So very extreme fasting in both cases. Okay, so marine heat wave. You guys may recall this marine heat wave, maybe, maybe not. It made a lot of headlines in 2014 and 15. The ocean in the North Pacific got really warm for a very long time, and a lot of things died, and a lot of things were getting washed up on shore where they didn't belong. Just all sorts of disturbance around the North Pacific. This is a composite of both 2014 and 15 in the spring and summer sea surface temperature anomaly to just give you a sense of the scale of both size and magnitude of this marine heat wave. It is one of the largest and longest and warmest on record. Not the top on any of those categories, but it was really massive compared to most marine heat waves. There were some areas of the North Pacific that reached over 5 degrees above normal temperature, which is very warm, very, very warm. Conveniently for us, we deployed 71 CTD tags during the years 2014 and 2017, and our seals swam right through the middle of this giant blob. So they were taking measurements of temperature and salinity down to whatever depths they were diving, usually about 800 meters, and these CTD casts were collected about every 6 hours, depending on how deep the seal was diving. If they dive really deep really often, then you actually get more CTD data than if they're diving more shallowly. The casts were collected at 1 hertz, so a measurement every second for the entire ascent of the animal. And I used all of these data along with climatology from World Ocean Atlas to calculate anomalies and do a whole bunch of interesting things that I'm going to show you with this salinity and temperature data to just try and characterize the 3-dimensional nature of this marine heat wave. Because at this point in time, the majority of the literature was about the surface, because that's what was easy to see, especially from satellites. The only subsurface papers looked at down to 300 meters. So there's actually a dotted line on these 2 graphs showing 300 meters as just an indicator of kind of where we had looked prior to me digging into this elephant seal data. So one of the first things I did as a comparison to the literature was I grabbed what has been sort of defined as this core area of the marine heat wave, which is everything inside this box. And I calculated average anomalies on a monthly scale to look at the evolution through time. So this figure starts in January of 2014 and goes all the way to the end of 2017, because I wanted to see, like, okay, the blob disappeared ostensibly in the surface in 2015. However, it got kind of complicated because there was an El Nino in 2016. So, you know, the ocean's not that simple. It didn't draw a lot. Sorry, there are lots of marine mammals right out the window right now, and that's very distracting. There's a whole group of something porpoising out there. Apologies, can't help myself. So there was an El Nino in 2016, which meant there was persistent warm anomalies happening, and I don't know, where one thing ends and the other begins is very ambiguous. So rather than, I was just interested in, like, where was it warm and how long was it warm for in this area of the ocean? So the top panel here is absolute temperature anomaly in degrees. So this very light color is like 0.2 degrees, so really small in terms of like total difference. The bottom scale, or the bottom panel in B, is normalized to the variance at that depth. So as I'm sure most of you know, when you get into the deep ocean, temperature does not vary very much. So a 0.2 degree change is actually a huge change at 800 meters of depth. So this dark red is showing between 3 and 4 standard deviations warmer than normal for that depth. So the big take-homes here were, first of all, there was substantial warming below the surface all the way into 2017, even well after sort of the surface appearance of the heat wave had disappeared. And then if we look below 300 meters, there were significant warm anomalies even down to 800 meters. Now, the thing is that this heat wave originally appeared as a result of surface warming, as a result of changes in wind patterns that caused essentially warm water to build up in the North Pacific Ocean. So how did that get down to 800 meters? Because that's a long way for warming to just penetrate vertically through the water column. So we dug a little bit deeper. Are you guys familiar with spice as a principle? So we took the combination of temperature and salinity data to actually look at density surfaces and look at spice anomalies on those density surfaces in order to try and figure out if there was some lateral movement happening at depth that might be contributing to this warm water where you don't expect it to be phenomenon. And we chose a couple of layers, essentially a layer right at the base of the pycnocline roughly, and then a layer deeper than that to try and kind of gauge some of what was happening. And I took, for every point on this grid, for every year, I looked for the nearest location in climatology that reflected the same spice, the same temperature salinity combination as that point did. So it's like, this is here, where could it have come from? And I did that for every point 1,000 times while adding some random variance to all the measurements to account for the fact that like our measurements are pretty accurate, but there is obviously some error in that. There's also some error in the climatology. And use that to calculate arrows to indicate where those spice anomalies might have come from. And it looks pretty consistently like there is northward movement from the subtropical gyre up into the region where there is essentially the gyre-gyre boundary. Elephant seals tend to use the gyre-gyre boundary as one of their main foraging grounds, so that's where all of our measurements tend to be. And it's consistent, especially in 2014 and 15. There's this very strong northward movement that's indicated by these spice anomalies. Moreover, we actually looked at the distance between these two density layers, and they got the thickness of this space between the two layers got thicker during those years, which would indicate water moving into that space laterally. So all of this is to say that we had really deep subsurface, we had really deep anomalies, deeper than expected. 2015 was really warm everywhere. That was where we had the warmest anomalies in both surface and at depth. And it is likely that there was lateral advection contributing to some of the subsurface structure of the marine heat wave in addition to the surface warming that was happening. And if you would like to know more, you can read a very dense paper that I'm quite proud of, but it is very dense. So fair warning on that one in terms of how we characterized this anomaly from the elephant seals. So then, the other fun part was looking at the behavior of the animals. I'm like, okay, there was obviously this warm anomaly. It got deep. Like, things were not normal, even down in the mesopelagic where elephant seals were foraging. What did it do to them, if anything? To do that, I used all of our elephant seal data from 2004 to 2020 and essentially created an elephant seal climatology by looking at all of the behavior we had from not anomalous years and comparing it to the behavior from those anomalous years. For my thesis, I use 2014 and 15 as anomalous years. Whether or not I should include 16 and 17 as well is kind of a question mark that I am still playing around with. This piece of the thesis is still a work in progress. It hasn't been published yet, and I'm reconsidering some of these things. We did, since those sort of groups of data represent very different sample sizes, there was a lot of either randomized subsampling or normalization that had to be done in order to make these measurements comparable. Excuse me. If anyone wonders about that, ask me afterwards. I don't get into the details here. So to start with, this is a normal elephant seal distribution. And when I say normal, this is a kernel density of all elephant seal tracks, excluding the marine heat wave years. So nearly 500 tracks of elephant seals. This is where they go on their 2 trips. The short post-breeding trip on the left, post-molting trip on the right. They use a lot of space. And in the post-molt trip, especially when they're out at sea for over 200 days, they're pretty dispersed. Like, there are not a lot of very clear areas where they are more than anywhere else, aside from they use this gyre-gyre boundary region between the subtropical and subarctic gyres. So 2014, 15, and 16 I included here. Can I get a water, perchance? I do not have one. If I could steal one, that would be great, because I have a feeling I'm going to cough some more. Is that okay?
Yeah.
Thanks. Sorry, I was not prepared. Apologies. So this is 2014, 15, and 16, and this is showing essentially the density distribution of animals during those years. And doing some subtraction, many thousands of times, we can actually see pretty clearly where there are increases and decreases in space use based on the density of the distribution of those animals. And like I said, there was subsampling and repetition in order to create this. It wasn't a simple subtraction from the 2 maps that you saw before, even though it looks that way functionally. Key things here are, in both seasons, the animals are using the subarctic gyre more, so they're further north. Post-breeding season, there's a lot more use of what I would call near shore for an elephant seal. It's not, excuse me, it's not really that near shore, but it's sort of near shore. And then a lot of use during the post-molt further out past the date line. I'm going to have to cough for a second. I apologize. Oh. Sorry. I just flew in from Japan today, so I'm a little, dried from the airplane, all sorts of things. It's been a day. So, in addition to space use, we also want to look at their diving behavior or depth use. So this is sort of similar to the first graph I showed you, except now we are looking at depth and time. So this is a conglomeration of all of the elephant seals and the depth that they dove to for all 6 million of their dives. That might not be a bad idea. Thank you. I don't know. Whatever works. Yep. Thanks, guys. Appreciate your patience. So yeah, this is standard elephant seal. They have some pretty distinct, thank you, depths that they like to dive to, driven by their prey. During the post-molting season, they tend to use 2 bands. This reflects a daytime and nighttime pattern that's related to the deal migration of some of their prey in the mesopelagic. During post-breeding, they're a little bit deeper, focused more deeper in general. So again, Did this change at all when we look at 2014 and 15? And the answer is yes, although it might be very subtle. I have stared at these for a very long time, and it's quite obvious to me what's different. So we will do a red-blue difference to see where we have increases and decreases in depth use. And there are two key things. During the post-breeding trip, they actually were diving more shallowly than normal. And during the post-breeding trip, during the post-molting trip, there's this really pronounced, very deep diving. And this is something that interests me a lot because it's very constrained in time in terms of when it happens and because it's just really deep. So this is why that pattern exists. So the upper left is a typical elephant seal, stereotypical post-molting elephant seal. They dive shallowly during the night until they hit winter, so during the summer and fall, and then they dive deeper around 600 meters during the daytime. And they'll have these like occasional deep excursions every once in a while. That's a very typical pattern. What's in the big panel on the bottom right is what I'm calling a deep dive pattern. And this is very different. Okay, the animals, while they are still doing their shallow nighttime diving during the daytime, in July, August, September, and summer, they are diving very deep, 700, 800, 900, 1,200 meters, and they are doing it consistently. This pattern is something that can be seen throughout our entire record, but it was more prevalent by far during 2014 and 15 than any other year before or since. Almost 2/3 of the seals exhibited some variation on this pattern, as opposed to like 1 or 2 here and there. So this is something that I'm curious about and digging into more. So there's a couple key things here. So elephant seals get really skinny during their fasts. Then they go back out to sea, catch lots of food, and start getting fat again. We can actually measure how much they're getting fatter by drift dives, in which the seals swim down for a while and then stop swimming and passively float. And when I say float, I mean glide either downwards or upwards, depending on if they are positively or negatively buoyant, which is a measure of their fat to lean tissue. So when they leave the colony, most of the time, they are negatively buoyant. When they return at the end of the post-molting trip, they are positively buoyant. So they have actually transitioned from negative to positive buoyancy. These deep dives are happening almost exclusively when they are negatively buoyant, as far as I can tell, with a giant asterisk that Arena, my lab mate, and I are redoing this analysis for a variety of reasons. However, we very much expect this pattern to hold. And the main reason for that is likely energy and efficiency of movement. If you are negatively buoyant, it is way easier to get down to 800 or 900 meters to find food. The downside is you then have to work hard to get back up to the surface. So there is a cost to it, but it does seem like once they get positively buoyant, it's just not worth the work to try and get down that deep. There could also be a seasonal effect, like maybe it's more interesting down there during the summer than it is during the fall and winter in terms of the prey that they can catch. Now what's interesting here is that one of the metrics of foraging effort that we use is wiggles. So if an elephant seal dives down, it then often does a bunch of these wiggles while it's foraging. Presumably more wiggles means more foraging effort. However, during this period, when they're doing all this deep diving, there is substantially reduced wiggling, which translates under our normal metrics to reduced foraging effort. And certainly it suggests that the prey field that they're exploiting during that time is different. Now there's been some work done with jaw motion accelerometers by some of our collaborators that has demonstrated that they do these very, very deep spike dives that have no wiggles in them, and as they're descending, their jaws are opening and closing repeatedly. So they are going sort of straight down, and whatever it is that they're going for, they just keep encountering on their way down. And they're either chasing something into the deep ocean, or they're going through a layer of something that they're able to grab multiple of. But this is a sort of a disconnect between actual foraging effort and wiggles. So that reduction in wiggles does not reflect a reduction in foraging effort, but it is, again, like they are exploiting a different type of prey and exhibiting a different behavioral strategy than they have in previous years. So some hypotheses about these giant deep spike dives that they do every once in a while is maybe they're going for larger prey items, maybe they're exploiting the low oxygen zones that are deeper in the ocean. These are sort of open questions that we are trying to figure out. I'm looking a little bit more closely at the period of time in which they're doing these deep dives and where they are spending more time during the marine heat wave. First of all, it happens that they are sort of like skirting the edges of the warmest part of the blob, like they're kind of outside of it more than they would normally have been. But in addition to that, those locations are areas where low oxygen zones are a bit shallower in terms of their depth of the ocean. So they may be more accessible and more readily used by those seals. So perhaps that is why we're seeing more of that deep diving behavior. If they are more able to pursue prey into low oxygen zone where the prey can't catch their breath, but the surface breathing elephant seal has no problem because it doesn't need to breathe down there. Yes, all of those things. So then, of course, the key last question, one of the key last questions is, did this matter in terms of their foraging success? And the answer is sort of yes. It seems like it has mattered. In particular, during 2014, their energy gain rate and their percent fat when they returned to shore were very high, much higher than they have ever been in any other season, especially for the post-breeding trip. Now here's the interesting thing. Their energy gain rate was high, their percent fat was high, but their mass gain was normal. So what does that means that there was something else that was traded off. If their fat gain was high and mass gain was normal, that means their lean tissue gain was low. So they gained less lean tissue than they normally would. So I said, again, they lose 25% of their lean tissue during their fasts. So when they go out to feed, they need both protein and fat. They need to rebuild both their muscle stores as well as their fat stores. So during these trips, they had an unusually high proportion of fat gain relative to muscle or lean tissue gain, I should say. And to be determined if that's actually a good thing. Like, is it, yeah, that's sort of a question mark. So yes, they had high success from an energy standpoint, but whether or not that's ultimately good for them from a body condition, long-term standpoint is kind of a different question. Yeah, and there was definitely a very marked decrease in energy gain rate looking from 14 to 15 to 16 to 17. Sort of in both seasons, although 2015 was just, post-breeding 2015 was weird. So this is also something that I want to look at a little bit more closely. So far I've been kind of looking at the anomaly as one block, but those years are not all the same. And it warrants sort of further investigation into the fine details. Not only was their energy gain rate, so like their daily acquisition of energy high, but their total energy was high. So it's not like they went on, it wasn't just because of their trip length that rate was unusual. Their total energy gain was just really high. They ate a lot of fat. Like they ate a lot of fatty things, especially in 2014. So revisiting this whole idea of like fishes versus squids, again, fishes are really fatty, squids are very not. Elephant seals will eat anything that they encounter. So something that I'm trying to do is think about their actual metabolic rate and foraging energetics to think like, okay, how many fish would you have to eat per day to gain the correct amount of mass to be an elephant seal? How many squid would you have to eat per day? And then how would that have shifted? And this is very general, obviously, but how would that have shifted to account for what we're seeing? I can tell you that you need to catch a lot of fish every day. Sorry, you need to catch a lot of fish every dive. So this graph is showing that you need to catch like between 10 and 15 fish every time you go down in order to make up the energy that you need as an elephant seal. Whereas you only need to catch between 1 and 2 squid every time that you go down. They have to eat constantly in order to maintain their metabolism and actually gain the mass that they need to gain. So, you know, they're, again, generalist predator, they're going to eat whatever they encounter. So a shift in the prey field that they're encountering could mean pretty substantial changes to their nutrition, essentially. They don't really have the luxury of going like, I don't think I'm going to take that fish. I'm going to take this fish. Like they've got to just take everything that they encounter. Oh yeah, they got to, yeah, 600 to 800. This is very ballpark, like back of the envelope calculation, but they got to catch 600 to 800 fish per day successfully in order to make a living. So key findings on behavior. They use the Gulf of Alaska more than they have in the past. There was a deep diving pattern during the post-molt that was very pronounced and may be related to the oxygen minimum zone, may be related to, drawing a blank now, so we'll leave it at oxygen minimum zone. And there's very likely been some sort of dietary shift or differences in the prey field that they're encountering based on the body composition data that we're seeing, along with some of these behavioral shifts that we're seeing. So maybe they encountered different prey because they were in slightly different space. That's entirely possible. Maybe they encountered different prey because just everything was different. Now, and their foraging success was either normal or high from an energy standpoint among the seals that survived. And that is a giant, giant asterisk because there was very high mortality during both 2014 and 15. So these are the ones that did well. We lost 25% of our animals to mortality in both 14 and 15, which is very unusual. It's more like 10% at most in most other years. We also had very high rates of skip breeding. So in addition to a bunch of animals not surviving, a bunch of females also did not reproduce after their post-molt foraging trip, especially in 2014. So this to me suggests that there was some sort of selective, like almost like a selective sweep happening, but there was selection for a specific, like this worked. This set of behavioral traits did okay during this marine heat wave. So rewinding, temperature anomalies at 800 meters of depth, peak anomalies in 2015, and probably some lateral advection contributing to the warm anomalies, and behavior, increased use of the north areas, so north of where the core warming occurred, and a lot more deep diving. Now, unanswered questions. And the thing is, I think you're probably not doing science right if you don't have a lot of unanswered questions. So these are a few of the unanswered questions that I am still trying to sort out and or want to try and figure out how to answer. Some of them are environmental, including, like, did the marine heat wave actually affect oxygen levels? Like, do we have any sense of how that worked, especially in the deeper ocean? Maybe the OMZ expanded a little bit in 2014 and 15 because of that warmer water. What's the time lag between a heat wave in the surface and actually affecting fish biomass at 400 to 800 meters of depth? Like, those things are not directly connected instantaneously. Are we talking months? Are we talking years? What the heck were they eating? That's one of my favorite questions. I'd really like to figure that out. Maybe we can find a way. Was deep diving a behavioral response, or is it an individual strategy that just happened to be successful and that those individuals always exhibit that strategy, but it's what allowed them to survive during this anomalous year. I think it's the latter. I'm not sure, but I think it's the latter. And then lots of questions about energetics especially, and this sort of weird body composition that we were seeing. And then, of course, fitness questions. So we do already know that weanling masses were a little bit low in 2014 and 15. Not the lowest we've ever seen, not catastrophically low, but definitely low. Pup survival to weaning was very low in 2014. And that could have nothing to do with foraging success and everything to do with storms hitting shore during the breeding season. So there's all sorts of different factors there. One thing we have not looked at yet, though, is whether those cohorts of pups have survived as well as other cohorts of pups have. So was there good food for the puppies when they headed out to sea to allow them to survive their first year? Or was it just normal? Yeah. Above, it could have been better, could have been worse, could have been normal. Only about 50% of elephant seals survived their first year. So it's a rough ocean for them. So all of these are still pending questions that I would love to answer. If anyone has any thoughts on any of them, please let me know. With that, I would, of course, like to thank all of the very many funding sources that have supported the work over the years, starting in 2004. And all of the people in Dan Costa's lab and the extended family that have contributed to collecting the data. It's been a major effort for everyone. And with that, are there any questions?
So you said only 50% of elephant seals survive their first year. Do you think that that's because they're bad foragers, or do you think it's more predation, or what do you think drives that?
So 50% of the pups-ish survive the first year. So a couple of things. It could be because they make very good shark snacks.
Yeah.
They're nice, or killer whale snacks. So I think it's probably, there is predation to a certain extent. The pups they get no education from older animals on how to be an elephant seal. Like, they are totally on their own, go out into the ocean, find some food, good luck.
Mm-hmm.
So some of it is probably the availability of prey that they can easily catch, which may not be the same as prey that are good for adults. We don't really know. We don't know what pups eat when they first start to forage.
Mm-hmm.
And some of it is probably predation, and they are naive little creatures going out into a big scary ocean. They're very, like, seem sweet and innocent for the most part when you encounter them prior to their first trip to sea, and then they come back like 6 months later and they're very cranky and aggressive. And clearly it has been an education for them being out in the open ocean. So
Cool.
a combination of those things.
Okay, cool. Thank you.
Kind of piggybacking off that a little bit, but you mentioned the male elephant seals often lose their tags. Could that also be a result of predation, or is there some sort of behavioral aspect that makes them lose the tags?
So there's two things. It's not just that they lose their tags, it's that they just don't come back. So male mortality rates are much higher than female mortality rates once you hit adulthood. So female elephant seals, if they can make it to like 7 or 8 years old, there's a really good chance that they're going to make it to 15, maybe even 18. Males make it to like 12, maybe 13 or 14 if they're very lucky. So their survival
the answer. So those animals that didn't come back. Okay. I was like, maybe I did. I was like, I don't remember because like they still transmit dive data.
Yeah.
And so like, where did they go offline? Were they in similar areas?
That's in the unanswered questions. So yeah, so the animals that went offline, some of them have transmitted diving data. I have not looked at it yet, and I have not looked at if there's any pattern to where they go offline. Sarah Keenly did a bit because 2014 and 15 were in her chapter where she was looking at female-male offlineness. There hasn't been any clear pattern to where females blink offline. And aside from the females that go offline within like 6 hours of the colony, which is very discouraging, and you're like, well, you got eaten by something. That's really unfortunate. I don't think that we have any good sense of when or where. But yeah, we need to look at the transmitted diving data. That's a whole other endeavor because it all has to get processed differently and all that kind of stuff. So I don't know yet. I might not have asked you, but I was like, oh, where did yeah.
Well, this one's coming up. I'll just ask quickly. How do like elephant seals forage? Like, are they visual forage? I mean, they're foraging downwards, dark and deep. Are they visual? Are they using odor cues of their prey? Tactile? I'm just curious.
Yeah. So that is an excellent question that we have a lovely paper that I would show you if I can. So they do have, so their eyes are very dark adapted, so they can see all kinds of light that we can't see down in the deep ocean. Bioluminescence is important for them finding prey. However, the biggest contributor, as far as we can tell, to their ability to forage is use of their whiskers.
Cool.
So this is a paper that if you're interested, I would recommend having a look at. It was written by one of our Japanese colleagues, Taiki Adachi, who I was just seeing like before today for me. So we've actually put video cameras on their cheeks to watch their whiskers and the prey as they approach them underwater. And they essentially whisk their whiskers forwards periodically while they're diving. And they only do it once they've gotten past certain depths, like into their foraging range. When they actually feel the movement from so the whiskers are detecting little tiny ripples in the water, essentially. They're detecting the wake of the fish that they are interested in catching. And when they detect a fish, you can see their whiskers all go forward and stay forward, and then they start to pursue whatever prey it is and gobble it up.
Amazing.
Yeah, and most of their prey, we found, are not bioluminescent. So bioluminescence is helpful, but it's not the key. It's not the key thing. Yep. Yeah.
So kind of continuing off the predator question chain from earlier. So in the blob, there was a pretty significant shift in the white shark population out in Monterey Bay. And did that have a significant impact on the elephant seal mortality rate?
That is a great question. I don't know the answer to that question yet. Would need to actually look. One of the challenges with white sharks is that often their presence doesn't actually align with most adult elephant seal presence. So they're much more likely to have an impact on the juvenile and pups than they are on the adults. Because like right now there are a lot of white sharks out. There have been for the last month and a half.
real fast. You were talking about how when the seals came back during that 2014, 2015 period, they were really fat, but they had very lean muscle. Less than normal. And you were kind of saying how that was an important factor in their fasting strategy. Did you see differences in their fasting strategies that year, or milk composition, or whatever else?
So potentially yes. So like we didn't measure milk composition.
Yeah.
So we didn't take milk samples. If only in retrospect we had taken milk samples. But the weaned pups that year, those years were lower in weight, which suggests that they got less milk transferred to them. It could be that the actual lactation duration was shorter. So research, actually, I think you and Crocker have done a lot of this work. Protein is really consistent in their milk across their entire lactation period. And Dan Crocker has found that probably one of the key triggers for ending lactation is when your lean tissue stores have gotten too low. Like that's the I gotta go now, I gotta get more food. It's not fat that is the limiting factor for them. Typically, it's actually their lean tissue stores. So if their lean tissue stores were lower, then yeah, there's a good chance that they shortened their lactation duration, even slightly. I mean, these pups are putting on like 8 kilos a day, so even just a 1-day shortening is a pretty substantial increase in energy transfer to the pup.
I have a quick question off, but it's just because
Yeah.
the computer is that, and it's related to that. So it's, do you think the change in body composition from more fat tissue to than lean tissue resulted in the females having a skippier, not being able to successfully carry a pregnancy to term and or wean a pup?
The short answer is yes, that is entirely possible. It's, as with everything, it's a little bit complicated. It's never as straightforward as this one thing caused this reproductive failure, but certainly given the amount of, given both the low survival and the number of skip breeders that we had during that year, I think it's likely that they were struggling to get the resources they needed in order to successfully reproduce. As capital breeders, elephant seals essentially they put on all their stores and then they give it all to their pup at once and then it's done. Theoretically, if you think about it, you should not have a pup if you don't have enough stores to wean an adequately sized pup. Otherwise, it's just really not a good use of your energy, right? Like if you can know physiologically that you have not enough to wean a pup that has a remote chance of surviving, then why would you spend the energy doing it at all, right? So there is some thought that, yeah, there is probably a physiological switch that goes at some point in the pregnancy, I don't have enough stores, we're going to turn this off. Or I do have enough stores, we should be okay. We should at least be okay. Maybe not great, but theoretically enough. So probably, but there are also other contributing factors like disease and, it's possible that a number of females actually don't implant not because of their post-molt gestational trip, but because of their prior trip didn't go very well. So there's a lot of different time points we have to check in order to really think about why did this female not breed.
I'm not a vertebrate person, so I know nothing, but I was really curious about the shift in prey that you were talking about during those anomalous years, and I was wondering how you would be able to trace that in the future. Could you use stable isotopes? What would you be doing to examine that?
So that is a great question. So stable isotopes, yes. We actually have stable isotopes from 14 and 15 and a bunch of years around it, and spoiler alert, they are not different at all in 2014 and 15. So we have looked at that. Only bulk though, so bulk carbon and nitrogen, which is not entirely surprising if they are foraging in similar locations and at a similar trophic level, finding that kind of change would be hard. The better way to
So just following from Sebastian's question earlier, so you said that for this, during those anomalous years, they have more lipids than they do have lean tissues. So in terms of the milk that they produce, I know you haven't measured that, but what do you think? Do you think there will be more fat content in the milk, or because they have so much of the fats, or do you think that remains pretty consistent throughout? You know, even it doesn't matter like what their body composition is, but the content remains the same.
Well, based on Gita's master's thesis, I could answer that question. Based on Gita's master's thesis, their milk composition stays, the protein part stays very consistent, and there's no real reason to expect that would be different in that year. It's possible, but it's more likely that they just then had more fat than usual at the end of lactation, you know, when they got to the point where their protein stores were depleted enough that they had to leave. When you consider this, like the pups are born at 35, 40 kilos, and they need to grow to preferably 150 kilos in 4 weeks. And not all of it can be fat. Like they need to actually grow in terms of somatic growth. They need to put on muscle. They need they need that protein. So, you know, I think that decreasing the protein content would not be a good strategy in terms of the success of that pup, because it's about much more than just energy. They need all of the stores that they need in order to go out, and they fast for about 6 weeks, 6 to 8 weeks after lactation ends and before going out to sea. So the pups also need pretty massive stores in order to make it. So I don't think so, but no, that's just my gut feeling on it. / I think Gita agrees with me, and she knows this better than I do.
Protein doesn't seem to change.
Yeah.
10%.
Anything else?
I'll preface this by saying I'm not familiar with marine mammals at all. Know nothing about tagging them, but you did mention tag size at the beginning of the talk, and I was wondering, kind of a two-parter. First, have the tags that you've been using been getting smaller, and is the data, like if they have been, is the data that you've been getting back, has behavioral patterns and foraging shifted at all?
I'm going to defer to one of Gita's grad students on this particular topic, who is she's present, but there will be a defense in the near future to discuss these things. Those are really good questions. Short answer is that yes, tags are getting smaller a little bit. The biggest tags are getting smaller, is I guess what I would say. There's always effort on the part of the manufacturers to make things as small as possible but still do what we need them to do. The major limiting factor is battery. So like you can only get so small before you can't actually record for long enough for it to be of use. I will not speak to the behavioral side of things. I'll let Kaylee answer that in the future. In a few months. But yeah, it's a very good question, because you know we are attaching something to a very hydrodynamically designed thing, and it's like a blocky thing that we are attaching in some cases. So it is an important thing to consider. Anything else?